just going over the terms right now?
Yeah, for right now.
Okay.
And then,
okay.
Okay, so gene, what did you,
For
from
gene,
reading it said it was like the basic unit of hereditary principles, or just kind of the basic unit of heredity.
Right. I ended up putting a definition, a pattern of chemicals within a cell that carries information about the qualities passed to a living thing from its parents. So that was like that was the Google definition that I got from it, because I wanted to check other sources too. And so that was one of those. So
and then
information passed
kind
on.
of an extension of that. It's made up of DNA, which contains the instructions to create protein sequences that'll create alleles.
Okay.
I don't know.
Yeah.
Maybe on that.
Yeah. No, that totally works too, because it's true.
Okay, and then chromosome set.
For chromosome set, I got. Hold on. I ended up just putting chromosome. Sorry.
Okay.
I think I skipped that one. Yeah, I skipped that one by accident.
It's fine. Chromosome set. From what I see from kind of the reading,
that's all I go. Just like
so we have the homologous chromosomes, which are like the same. It's kind of similar to that, but kind of like that pairing.
Oh, yeah.
I just literally said funny videos.
Let's see. Because they're homologous chromosomes, diploids have 2 copies of every chromosome. In stomatics, so hold on. Okay, so kind of, this is what it said from my notes. Humans have 1 set from a father and then 1 set from a mom. So we kind of remember how we have those homologous chromosomes. We have the 2, right?
Yes.
You have 1. I think a set is just kind of the 1 by itself. It's kind of that individual chromosome. Let's see.
Okay. And then, so homologous chromosome just basically means the same chromosome.
Yeah.
Yeah. And then sister chromatids.
They're similar in size and
They're
DNA.
basically identical. Yeah, so they're identical DNA. And an allele.
There are some variations within the to create the different alleles. They are very similar. Let's see. Sister chromatids. It's kind of having 2 homologous chromosomes next to each other, isn't that? To create that tetrad? Or am I wrong? Oh wait, no. That's
so it says each individual gene within a pair is called an. Oh wait, where am I?
No, that's wrong. What I was saying about sister chromatids.
Let me see, sister.
I'm trying to remember where I put it though.
It says.
Okay, so sister chromatids are made during the S phase of the cell cycle, and it's a copy pair of the chromosomes. So it's sister chromatids are exactly identical, while homologous chromosomes are pretty identical, but they're not exactly.
Sister chromatids are like clones of each other, basically.
Yeah.
Okay. Yeah, so it occurs during the S phase, during anaphase 1. That's all I put for that one. And And then we
Wait,
have,
did you say of anaphase? That doesn't make sense because the S phase happens before mitosis or meiosis. Hello? Oh, I think you got lost. What? Hello?
yeah. Can you still hear me?
Yeah, I can hear you now.
Oh, yeah. Okay. Sorry. I was.
You kind of got frozen for a second, but
Okay.
as I was saying, you said that it was part of close to anaphase.
Yeah. So anaphase 1, because that's where I saw. Well, maybe that's what it says. During anaphase 1, the members of a homologous chromosome pair separate from each other towards opposite spindle poles. At this point, each chromosome will consist of 2 sister chromatids. Oh, wait, maybe I think.
Because the sister chromatids are made during S phase. So if you kind of think about the cell, there's different phases of it. When it's you have the G1 phase, then you have the S phase, and the G2, and then you have mitosis or meiosis, depending on the cell.
I just found it. I was looking at the at the wrong section, but yeah, I just found it. Overview of meiosis. Got it.
Yeah, I just wanted to make sure you're up to date on that because it's like because the S phase is way before the mitosis.
Right. Yes. Okay, sorry.
Just
I haven't
wanted to make
taken
sure
biology.
you're clear on that.
Yeah.
See.
And then we have allele. Okay.
Okay, so allele.
So.
Okay, so you have a certain. To me, it's like when you have a certain characteristic, like height or hair color. Allele is kind of the variations you can have of that characteristic.
Okay, so they just contain different genetic information that compose your characteristics, like physical and genetic, I guess.
Yeah.
Well, I don't know how to make sense, but okay.
Well, you're kind of thinking about the phenotypical and the genotypical kind of idea.
Yeah. And
because there are
then
going to be some variations between the two, as we see later, and some. I kind of read through some of the problems.
Okay.
But, yeah. Let's see. See. Homozygous.
Homozygous means same, right?
Yeah, so they'll have the same 2 alleles for a characteristic.
And then heterozygous.
Be different.
And
then dominant.
Dominant is the one that is expressed, and recessive is the one that isn't expressed.
Right? I don't know. I don't. I guess. I think. Because it is expressed recessive, it just has to have both recessive genes present, alleles present. But it's not. It's going to be under. It's going to get masked by the dominant when the dominant is present.
Okay. When the alleles are not identical, one allele is dominant over the other. Recessive allele. Okay.
Okay. Genotype.
Genotype, we would be talking about the. I'm trying to summarize, but like for instance, if you were to have 2 letters to say like green color, like green colored eyes, like G would be green and little g would be brown. If that, I don't know what to call it. That would be the genotype, like CG or GG or capital G, capital G.
Yeah.
And then
I kind of put it as the genetic classification of a a characteristic. So you kind of have that overview of like the different like letters that symbolize the different alleles.
Okay.
Yeah.
And then the phenotype is the observable characteristics of the organism.
Yep.
Okay. And then for 2 and 3, those are just like drawings, right?
Yeah.
And those we can just get off like the internet, or do we have to actually draw them and copy and paste them?
I think we do have to draw. I mean, I kind of, if you kind of turn back to me, I did kind of start doing that.
Uh-huh.
Let's see.
Okay.
Hold on.
Yeah. I see it.
Yeah, and then I kind of just put them in different places in my notes depending on what I was talking about. Because that was when I was just talking about phase 1. And then I got metaphase and then metaphase. And then anaphase, telophase. So yeah.
Okay, I didn't do that. I didn't understand the question, I guess. But I did not do a drawing, so I just Googled pictures of each one. Like,
And there
more
are
details because I
pictures
can't
in the
draw.
PowerPoint.
Okay.
Yes.
So
Okay, so I kind of did that for 2 and 3. Chimpanzees. So do you want to go to question 4?
yeah, let's do that.
Okay. So in chimpanzees, the diploid chromosome number is 48. For each of the following, identify the total number of chromosomes, the number of homologous chromosome pairs, and the number of sister chromatids present in each cell.
Okay, hold on. This is kind of playing around whether we know our terminologies or not.
I hung up!
Okay, so homologous chromosome. 2 copies of a chromosome. So diploid chromosome number is 48. And identify the total number of chromosomes, the number of homologous chromosomes.
So there would be 24 homologous chromosomes.
Yeah.
Right?
And then 48 total number of chromosomes. And I don't think, and there's no sister chromatid.
You look at it. God's word teaches us why.
And the number of sister chromosomes. Okay.
The word operates.
Okay.
Word operates.
Oh my gosh!
So do you want, so was that it for that one, or do we have to
You have to do one for each of them. See how there's like prior to replication, and then the beginning of metaphase, the end of telophase 1 and telophase 2, and after fertilization. So I think we have to do each one of those.
Okay.
48, 24.
So would it be 48, 24, the beginning of metaphase? 48, 24, and then 96?
For the first one?
For the beginning of metaphase 1?
Because we, beginning of metaphase 1, I'm just trying to think what we do. We have them all set up. Chromosomes are condensed.
Did you do it or not, babe? You did it, then mark it complete.
Okay, so I think we're still at the beginning of metaphase 1. I can't remember. I think replication does occur.
Right.
So I think everything's going to be doubled.
Right. Because, okay, so we have 48 total chromosomes.
Uh-huh.
And so that's what I was thinking. So 48 total chromosomes, 24 homologous chromosomes. And then those 24, since they keep multiplying, so would it be like 24, 48, and then that 48 doubles again into 96. Let me see if I can find something on my book because, or I also have a PowerPoint. I don't know why I haven't pulled that up. Oh, there it is. Okay. Because that's what I think is for the beginning of metaphase 1. I don't know if I'm right or not. I don't think I am.
Ready
Okay.
to file with your.
Okay.
Make them stop.
Because I think
Yeah.
Work in an hour? What? Why? Oh. Jonah, say no, quit.
Okay. Yeah, that's what I think is the answer for that. I don't know how to explain. I'm really bad at explaining things. So I'm really sorry. I'm probably the worst person to be in a group with.
okay, so I think oh, hold on. I'm sorry, I wasn't quite. So you were saying that so it's 46 and
48.
48.
48 total chromosomes, 24 homologous chromosomes, and then 96 sister chromatids for the beginning of metaphase 1.
Metaphase 1. I think it's actually hold on. Let me look at this. Sometimes I get so miscombobulated. Okay, so you had 48 diploid. I get so confused with this part. It's just like, what?
Let me see if I can find something to see if it makes it easier.
But I think you're right. We're gonna have we're gonna have 48 chromosomes, and then I don't know about sister chromatids though. I think that comes in later. Because I don't think sister chromatids don't come in until meiosis 2.
Okay.
So I think we're just gonna have 0 until we get to that part.
Okay. All right, let's just go to the next one then.
Okay.
Okay. So for the end of telophase 1, I got 24 total chromosomes, 0 homologous, and I don't know about the sister chromatids. Would it just be?
I don't think we're gonna have sister chromatids until we get to meiosis 2.
Okay. Okay, but for the first parts then, would you say those would be correct? 24 total chromosomes, 0 homologous?
I think so.
Or did you get a different? Okay. All right. And then the end of telophase 2.
Just
I don't know if I'm doing this right.
yeah, I know. I'm like,
Okay.
They're called jazz hands.
Lots jazz hands.
oh, I was wrong about the sister chromatids though. I think I kind of was looking back at it. So, This is where we're going to have, hold on. I'm looking at the wrong
Okay.
thing. I swear there's so many tabs. Okay. Okay, so I think it's actually, I think you're going to have, well, in the end of telophase 2, you're going to have 24 in each of the cells. 24 chromatids.
Mm-hmm.
So you're probably going to have 24, so you're going to have 12 homologous pairs. Wait, no, you're not going to have any homologous pairs. You're going to have, I don't know, hold on.
Are we at the end of telophase 1? Is that the one?
Telophase 2, aren't we on telophase 2?
Oh, okay. Got it. Let me just scroll down. Okay. The end of telophase 2. Yeah, so 24 total chromosomes.
And then I think that's it, because we shouldn't have any pairings, because these are the haploid cells.
Right.
And then after fertilization, it comes back to its complete and full form of 48 chromosomes.
Okay. Mm. Yeah, that seems about right. I'm Googling it right now, and it seems right.
Yeah.
Yeah. Yeah. Okay. And then after fertilization, we would have 48, 24, and then I got 0.
Yeah.
Okay. Okay. For 5, suppose you're examining the plant height characteristic in pea plants. Plant height is controlled by 2 alleles. Capital T, tall, which is dominant, lowercase t, dwarf, which is recessive. In a cross between heterozygous plants, tall pea plant and a dwarf plant predict the genotype and phenotype ratios of the offspring. Okay.
This is a pretty easy monohybrid cross, or mono.
Just, okay.
Yeah.
I don't know where my notebook is. Okay. So we just have to draw that.
I think we can just say what the ratio is.
All right. Let me do that really quick. Sorry.
No, you're good.
So plant height is controlled in a cross between a heterozygous tall T and a dwarf plant TT. So TT, small TT. So it'd be 50-50.
Hold on. Yeah, it would be 50-50.
Okay.
Because you would have two that are heterozygous, and then you'd have 2 that are recessive.
Right. So this is the one I made. I don't know if you can see it, but yeah, there we go. This one.
Yeah.
Yeah, so those monohybrids I like.
Yeah.
Okay.
And then for the phenotype, we have 2 that are tall and 2 that are dwarf.
Right. Okay, 2 tall, 2 dwarf. Okay. We're almost done. Number 6. Let's consider the following characteristics in pea plants. Trait 1. So, what are the predicted phenotypes of the offspring? that has constricted pond. Across maybe between pond that has constricted. Oh.
Where are your quarters too?
If I let you get my quarters.
Yeah. Well, she kind of like I said. I was thinking like 5 days, and she was like, "Oh well," and I was like, "Not my quarters," and she's like, "I really want my quarters too." I was like, "Okay." I'm gonna sleep on that floor. I'm a good person. Just letting you know. I could have kept them. I could have been selfish. You could have. No, selfish.
That's best. And this is interesting. Y
You're such a jerk. You're supposed to be grounded. Come help me.
times consider that S and Y.
Okay, so let's see. Phenotypical.
Firstly, I kind of get the idea on how to make the hybrid table, but I don't know how to organize it. If that makes basically, it's just like the other one, except this time it has four across and
Yeah.
four
Well, I
going
kind of
down, right?
simplified mine a little bit, and since I knew some of the traits would be the exact same, you can kind of look at mine. It's kind of light.
Okay.
See how I kind of simplified
Okay.
it? I do have these
Yeah.
other traits here, but I know that they're going to be the same. So I can kind of double it. And let's see. So I'm probably going to have, so let's see. So 1/8. Hold on. I kind of have to work this a little bit more.
Okay.
So I'm going to have like 2 of the same thing. So of each, so they want us to do the ratio of the phenotypical things. So my capital S stands for, or like my cursive S stands for, hold on, I gotta look this thing, for smooth. So I have 1, 2, 3, 4. And then let's see. So I have 1, 2, 3. So I have 6 that are going to have the smooth, which are going to be smooth yellow.
Okay.
And then I'm going to have 6 that are going to be, and then I'm going to have 6 that are going to be constricted yellow. And then I'm going to have, yeah, I did that one already. And then I'm going to have 2 that are going to be smooth green.
Okay.
And then I'm going to have 2 that are going to be constricted green, which gives us a total of 16, which is how many you should have in your thing. So this work type setup. Does that kind of make sense?
Yeah, it does. It's just I'm having a hard time drawing it.
Mm-hmm. Yeah.
But no, it does make sense. I just remember a while back ago, there was a classmate that showed us, he didn't really use a table as much. He used something. I just can't remember what it was. I was
You
trying to
probably
see.
use the fork method, which they talked about in the reading.
Is that on the PowerPoint as well? Or is it
Not
just on
very
the...
well. It didn't really show it. It kind of talked about it, but it didn't show how to do it. Which is, I'm like, really?
Yeah, it just shows a graph or a diagram. Okay. Okay.
Which is probably
So
what we should use for the next one.
Mm-hmm. Okay, so the ratio for this one, so you said it would be half of each. So 16. So would it be, you said 1/8 earlier, right? Would that 1/8 be smooth yellow? Or is that smooth green?
Wait, we're just trying to predict all the
Just the
what all
phenotypes?
the phenotypes would be.
Oh, so
Like,
we don't have to give the genome.
yeah, so we can say 2 constricted green, 6 smooth yellow, 6 constricted yellow, and then 2 smooth green.
Oh, okay. All right.
The next one, number 7, is when we have to be more specific.
Okay. Okay, so 7. Suppose you can perform the following cross. A, B, C, D times with little a, B, C, D. Assume that all of the genes involved undergo independent assortment. What proportion of the offspring is expected to have the A? So heterozygous A, heterozygous B, homozygous C, and homozygous D genotype. Calculation of proportion. The fork line method. Okay, I don't really understand that method as well.
Okay, so this is how I would go about to do it. So you kind of separate them into like their own little monohybrid crosses. So you have big A and little a crossed with little a. And how much would we get to? We'd have 1/2 of, it'd be a half ratio. So we have 1/2. And then we'll times that by whatever. So to get the big B, little b, we would create another half. And then to get little c, we have to times it by 1/4. Or we could get 1/4 of the cross. And then big D would be another 1/4 because it's in that one corner. So if you times the bottom together, you get 1/64.
Okay, that makes sense. Okay, the way you explained it just now, that was perfect. That made me understand a lot more. But yeah, okay, so you just got those pairings and then just multiplied them together and got 1/64.
Yeah, 1 divided by 64. I kind of just took them into each individual monohybrid crosses and then mashed them all together.
Yeah, okay, that makes it simple.
I did that in a different class, so I was just like, okay, I liked it.
Yeah, because that's what I'm doing right now. And it's like, oh my gosh, I get it now.
Yeah.
Okay.
Because with a cross like this, you would have to have, and to create like a Punnett square, you would have to have 260 something squares.
Right. Yeah, it would be very long.
Oh, you'd have 256 squares.
Oh my gosh. Okay. D. And then the other one was DD. Okay. D. Okay, sweet.
Yeah, and I think that's it.
Awesome. Oh my gosh. Well, this was different.
What?
This was different.
Oh
Like
yeah,
doing a
I had to
visual
do this
presentation.
for another one of my classes, like this online thing. So I'm pretty, like, kind
Knowledgeable
of
with
more
this.
knowledgeable. More.
Yeah,
I wouldn't say I'm like
but
expert.
expert. No,
I'm
but
like here, and you're
yeah.
probably like here.
Yeah, you have the gist of this. I had no clue. I was ready to drop yesterday, but my mom was like, "Don't drop it." I'm like, but it's so difficult because I have a fixed mind, I guess, where if I don't get it, I just think, okay, I'm not going to get the entire course. But this was easing me through.
Yeah.
Yeah, so this is good. Okay.
Well, good luck.
Thank you.